Hello, everyone. My name is Annalisa Scimemi, and it's my pleasure to introduce this session of speakers. Just a quick reminder, add your questions to Crowdcast, and if there's any question that remains unanswered, please go to Neurostar and type it there. Our first featured talk for the day is from Adriano Bellotti from the Department of Engineering at the University of Cambridge. The title is Neuronal Morphology imposes a trade-off between stability, accuracy, and efficiency of synaptic scaling. Adriano, take it away.
Thank you so much, Annalisa. Hello, everyone. My name is Adriano Bellotti, and I'm a graduate student at the University of Cambridge in England. Today, I'll be talking to you about our project from the control group in the Department of Engineering. The title of my talk is Neuronal Morphology imposes a trade-off between stability, accuracy, and efficiency of synaptic scaling. I'll be showing you some of the theory, the model, and simulations from this project. So let's get started. We'll begin with some background in neurobiology, specifically the mechanism that forms the foundation of learning and memory. We'll start by defining what a synapse is here. A synapse is a junction between two neurons. It allows a presynaptic neuron, this bulb-like structure here, to pass an electrical signal to the postsynaptic neuron, the long blue projection. Synaptic potentiation is when a synapse becomes stronger, so the strength of this connection gets stronger. Hebb's postulate is a foundational theory in neuroscience that says neurons that fire together wire together, and this holds true here. Increased activity in the pre- or postsynaptic terminals strengthens the synapse. I'm representing that here with the thickness of the borders, so the synapses get stronger over time. Now, this happens at the level of the individual synapses, but in the context of cells and neural circuits, this is what enables us to learn and to retain memories. But this kind of feedback is dangerous. It can firstly result in saturation of input signals, where the overall strength of the cell is saturated. It can also result in a sort of vicious cycle. Correlated activity between neurons leads to synaptic potentiation, and synaptic potentiation leads to more activity. So what keeps these in check? What keeps this from strengthening forever? What balances the books and prevents oversaturation? Let's take a look at this in the context of a larger cell. So in this example, we have a larger dendritic tree, and a single branch is being potentiated. Just below the neuron cartoon here, we're tracking neuron activity, which is essentially the number of times the neuron fires an action potential. And that's experimentally correlated with calcium concentration, which is the curve at the bottom. Now, when a branch is potentiated, we expect the total neuron activity to increase. We have more firing in the middle plot, and calcium concentration increases, as we expect. How does the cell prevent this from continuously increasing over time when more branches are potentiated? If left unchecked, this could result in excitotoxicity at the cellular level, or at the level of the brain might manifest as seizures. The neuron's solution is to globally decrease its total excitability, to decrease the strength of all its synapses. This is called synaptic scaling. Synaptic scaling has been proposed to do a lot of things that I've listed at the top right here. It is thought that synaptic scaling prevents the vicious cycle that leads to unconstrained excitation. Synaptic scaling also maintains the relative strengths of the synapses. After synaptic scaling, this last step here, notice that the potentiated synapse is still stronger than the rest. That change was preserved. This is why it's integral to learning and memory. Stable, reliable changes at the molecular cellular level, like this result, in stable, reliable memories. So this is what synaptic scaling does and why it's important for our cognitive functioning. But this begs the question, how does it work? How can a large dendritic tree with many branches all coordinate to reduce synaptic strengths equally? Here's a neuron expresses some AMPA receptor, shown at the bottom.
Adriano,
These receptors shape electrical inputs. Oh, I'll start from the beginning of the slide. This is a figure from a prominent review that provides. Good? Okay. That provides an answer. This figure shows that a neuron expresses some AMPA receptors, shown at the bottom here. These receptors shape electrical inputs in the cell, which are shown here, which change neuron firing and change internal calcium concentration in the cell. This is regulated in the nucleus of the cell which sends out scaling factors and other signals that regulate AMPA expression. These scaling factors can be AMPA receptors themselves or others like ARC protein, shown here. The takeaway here is that synaptic scaling requires global regulation of AMPA receptors. This is kind of nice, a seemingly difficult problem with a large dendritic tree, much more complex than any diagram I've shown so far, and the cell needs to normalize all synapses by the same factor. The only way to do that is with global control. But this brings up a few questions. How does the global signal work? How does it get transported everywhere? Why is it so slow? We observe settling times for synaptic scaling of 24 to 48 hours. Does it matter that it's slow? What influences does cell morphology have? Are there inherent fundamental constraints because of this? To address these questions, we've built a model. Our model contains realistic AMPA receptor production, transport, insertion into synapses, as well as global and local feedback based on calcium concentrations, all of which are consistent with experiments and the leading hypotheses for synaptic scaling. So this model so far has produced some nice findings. The global control required for synaptic scaling is a limitation for a few reasons. First, neurons are pretty big, and transport can take a while, especially in dendrites that are up to 1 millimeter in length. 1 millimeter in length. This results in a phase lag, or a lack of synchronization between what's happening in the center of the cell and what's happening with the AMPA receptor at the ends of the cell. And this asynchronization can be a source of instability. As a familiar example of this, you can think of driving a U-Haul truck or a boat or any other heavy vehicle that has a lot of weight and inertia. When you turn the wheel, say, to go to the right, there's a bit of a delay before the vehicle actually turns right. That's kind of what phase lag is like. There's a time delay between turning the wheel of the heavy vehicle and the heavy vehicle turning, just like there's a time delay between changing the scaling factor and neuron activity changing in the far ends of the cell. If the time delay is too big, you might lose control of the vehicle or miss a turn, just like the neuron might misregulate and create an instability. This explains why synaptic scaling is slow. It can take a day or two because of the phase lag. This limitation also imposes a trade-off. Synaptic scaling of the neuron is a balance between efficiency, accuracy, and stability. But before we get into the results, let's dive a little deeper into the model. So this is the model. Let's move our neuron up and break down all the individual parts. We're interested in neurons with a soma and a large branching dendritic arbor. If we zoom into the soma, we can see the site of cargo production and global feedback. Our cargo of interest is AMPA receptors, which are these dark blue dots. And we're using AMPA receptors themselves as our scaling factor. They're produced in the nucleus and transported on microtubules, these light red tracks. They're then released for spread throughout the cell. Global regulation occurs based on the total AMPA receptors on the surface of the dendrites. If neuron activity is too low, more AMPA receptors will be produced. If neuron activity is too high, AMPA receptors production will slow down. Next, let's zoom into a dendritic branch to see what transport looks like. Here we see that inactive AMPA receptors on the inside of the cell are labeled M, while those that are active on the surface are labeled G. Internal receptors M are transported on the microtubules with forward rate VF and backward rate VB. Synaptic sites are labeled with C, and there must be an available synaptic site on the membrane for activation of an AMPA receptor. C is the capacity of the synapse. In other words, C is the number of available slots for AMPA receptors. The C, or capacity of a synapse, is important for long-term potentiation and depression. We can impose an abrupt change in C to emulate LTP or LTD. In fact, this is what happens physiologically. A major postsynaptic change during LTP is an increase in structural proteins that support AMPA receptors on the membrane. Lastly, if we zoom into the membrane itself, we could see what cargo insertion looks like. Cargo is activated from M to G with some rate, activation rate, SI, and cargo is inactivated from G to M with some inactivation rate, S minus I. This receptors in a local environment regulates SI, but there's more on that later. It's worth noting that this model isn't just conceptual. We aim to make it data-driven. Some of the quantities can be broadly quantified from experiments, like the velocities, the insertion speeds, and the degradation rates. We can make conservative estimates of these and keep them fixed. Using these numbers, we've made a coarse-grained assumption with as few free parameters as possible. So this is a kind of visual abstract for a model. And using these components alone, we can describe the limitations of synaptic scaling. And that's what I'm going to show next. This first simulation validates synaptic scaling and introduces its limitations. We have a full-size pure metal neuron, which is approximately 1,000 microns or 1 millimeter in length. And we're going to simulate the production, distribution, and regulation of AMPA receptors. And again, this has been calibrated to actual experimental measurements. 5 hours into the time course, we're going to simulate synaptic potentiation in the distal synapse. These red compartments here are being potentiated. In real brain anatomy, this might replicate the perforant pathway, which runs from the entorhinal cortex layer 2 to the apical dendrites of the CA1 hippocampus. And again, 5 hours, synapses experience, the synapses in red experience a doubling of synaptic strength. The red compartments are undergoing LTP. In the curve at the bottom, each of these lines represents the density of AMPA receptors in one compartment. The nice result here is that scaling works solely with a global receptor. So this is a bit of a validation. The red compartments are scaled up, as expected. The unpotentiated black compartments are scaled down, which is notable because we didn't touch these. They went down on their own when the others were potentiated. The relative weights of the synapses here are preserved. We can visualize this with a video. At the top here, we have a trace of calcium concentration. Remember that this corresponds to global feedback, and we use it here to approximate the total activity of the cell. You can see that potentiation perturbs the system a little bit, but it seems to recover. Notice that at the end, the distal synapses down here have increased AMPA density compared to the proximal synapses. Now, this is a nice example because it shows all three possible limitations of synaptic scaling, and let me show you what I mean by that. To orient us through these limitations, let's lay them out on a triangle. We can summarize the trade-offs for synaptic scaling as a balance between three properties: efficiency, stability, and accuracy. As a preliminary definition of these three properties, let's say that stability manifests itself as overshoots and oscillations. This is a slight oscillation up here. The neuron slightly overshoots its target before resuming its steady state. Accuracy is the maintenance of synaptic ratios while scaling. Notice that after potentiation, the synaptic strengths are more spread out than they were before. This is inaccuracy. The synaptic ratios are not perfectly maintained here, so that's also an inaccuracy there as well. Efficiency is the speed or time constant with which we reach a new steady state. This is a measure of how quickly stuff gets to its destination, how aggressively a neuron responds to change. This quality of synaptic scaling with these limitations is the best the cell can achieve. For example, to improve the efficiency, the accuracy or stability is going to take a hit. To improve any attribute, one or both of the others will worsen. And the morphology of the cell itself imposes these restrictions. We're going to talk about how a neuron might navigate these trade-offs, which requires going back to the model. As we talk about the different parts of the model, I'll use this key at the top here, and I'll emphasize the parts that we're talking about. So let's begin with global feedback and cargo production. A cell that requires higher efficiency, faster production of cargo, might have increased sensitivity of global feedback. In neurobiology, global homeostasis is a cell's way of maintaining its level of excitability. This prevents overexcitation of synapses, called excitotoxicity, which can be harmful to the cell. The goal here is to approximate total activity in the cell. The sum of all AMPA receptors, G, produces an active membrane potential, V. The membrane potential then modulates the amount of internal calcium. And with calcium concentration, we can compute a global error from some target calcium set point. That's the first equation I've listed here. And lastly, from this global error, we have a production term U. U is the rate of biosynthesis in the soma. What we've set up here with U is called a leaky integrator. With each time step, the error is integrated to give production term U. Kg here is the gain or the amplitude of global feedback. This Kg, this gain, determines the sensitivity of the global controller. This second equation with U represents the dynamics of gene expression with regulatory feedback. See? Global regulation mediates the trade-off between efficiency and stability. Let's see what this looks like in a full morphology. Good connection? Yeah.
Yeah, connection is sometimes, but okay.
Okay. We're going to simulate with weak global regulation and strong global regulation. All other parameters are constant here. We're going to sweep through a series of global gains, or kg, as a free parameter. And this is the result. With weak global regulation on the left, we have a stable but slow system. If we increase kg in the middle, we can speed up cell responsiveness, but we become less stable. If we further increase kg on the right, the neuron fills very fast and is very unstable with oscillations. From this trade-off, we clearly see that there's a hard limit to how quickly a neuron can respond before we see oscillations. So what if the neuron needs to fill as fast as in this middle case, but with less oscillations? The free parameter that we were changing here, remember, was global feedback, in which all compartments change the total activity of the cell. A neuron can also improve stability by increasing regulation at the local level, where each compartment regulates its own excitability. And we'll see that next. So let's look at local feedback. A few months back, my supervisor asked me to design a mechanism for local AMPA regulation. I started digging and I found this figure for local AMPA regulation, and I started realizing how complex it was. There's something like 20 molecules here and many, many pathways. I realized that although it seems very complex, what's going on here can be summarized with movement in two directions. Some AMPA receptors are activated, we're calling that SI, and some AMPA receptors are inactivated, that's S minus I. So that's what we did. That's how we designed it. And this entire figure outlined in green can kind of be simplified to one arrow for AMPA, for local AMPA regulation. In the discretized compartmental model, it looks like this, where the amount of active cargo GI regulates the amount of cargo activation, SI. Mathematically, we again have the same target calcium concentration. If there's too much local excitability, SI decreases. If there's too little local activity, SI increases. So local control imposes a trade-off, this time between stability and accuracy. And we're again going to show 3 simulations, this time with increasing local control. So we're going to have weak control here and strong local regulation on the right. With weak local regulation on the left, we have accurate scaling, where the red compartments see a notable increase over the black. However, there's poor stability with the same oscillations that we saw last time. In the middle, the oscillations are improved, but the difference between red and black synapses begins to diminish. On the right, we have the strongest local regulation. Synaptic scaling here is no longer accurate. We get a minimal increase in red synapses compared to the black, but the system is fully stable now. We can think of synaptic accuracy as how reliable a stored memory is. In the left and middle cases, this memory is still registered. In the right case, a cell can barely distinguish between those synapses, and the distinction is lost. So let's go back to this slide to reorient ourselves. So far, we've shown that global control improves speed at the cost of stability, and local control improves stability at the cost of accuracy. Now, how might a neuron prioritize accuracy? This brings up the efficiency-accuracy trade-off, and this is mediated by internal pool size. So let's go back to the model to see how this one works. So by internal pool, we mean the amount of cargo in the inactive, or M, state. In the model, the free parameter now is this S minus I. On the left, we have a large S minus I and a large internal pool of AMPA receptors. On the right, we have a smaller S minus I and less available inactive AMPA receptors. And this result is a simple consequence of mass action. We have many more M here than we do on the right. So what's the point of this? What difference does this make? Let's take a look at the third trade-off. So now we're going to be varying this S minus I parameter, which corresponds to the internal pool. And as before, all other parameters are going to be held constant. On the left, we have a small internal pool. Notice that this fills relatively fast. In the middle, we have a larger internal pool. We can begin to differentiate between potentiated synapses in red and those in black. However, the cell fill time is slower, less efficient than it was on the left. On the right, we have the largest internal pool, and we can see pretty good distinction between red and black compartments. But notice how much slower it is. We have better accuracy, but it becomes almost unbearably slow, also notable here in the calcium concentration. So by now, we've seen all 3 trade-offs, and we've seen examples of how to navigate free parameters that navigate this trade-off. We have our model overview here, And by tweaking the global regulation, local regulation, and internal pool size, we can explore all possible scenarios for these three attributes. So these are where they fit in the model overview. So far, I've shown you a bunch of simulations, all in the same model neuron. And I've just been talking about this triangle. But why should you guys be listening to me? How do we know that what I've shown so far generalizes to all neurons or other morphologies? If we scale down the complexity of neuron morphology and boil away all the extraneous parameters, are we still left with these relevant dynamics, with this trade-off here? And how do we know that there's a fundamental underlying relationship here? At the same time, we want to rigorously quantify these trade-offs to understand how these attributes are connected and how they balance. We essentially want a theory that connects all these, and we're looking for the tools that we might need for this. Thankfully, there's a few ways of doing that, and this is where my collaborators Saeed and Fulvio helped. They developed and pushed this theory in the context of neurons. And this is a collaborative effort, so I'll be showing my take on the work that we did. So let's begin with the theory of stability and how we might rigorously quantify stability. We have a fairly complex system with a lot of moving parts, and we can simulate our system. This is what we've been doing. And one of the parameters that modulates stability was global gain, or kg. This is what we saw with the stability efficiency trade-off. As we increase kg going down this way, the cell fills faster, but also becomes less stable. And we begin to see overshoots and oscillations, like in the purple trace in C4. How can we quantify these levels of stability? We're going to do that with Nyquist stability criterion, and I'll walk us through it. First, we're going to approximate our system with a block diagram. This diagram has an input, an output, a system block G, and a control block C. We can conceptually define an instability as when the output is very, very large compared to the input. In other words, when the ratio of output to input tends toward infinity. This output to input ratio is also known as the transfer function, which some of you may be familiar with. We can then solve an equation for output over input. Using the block diagram, this transfer function is equal to G over 1 plus GC. And again, we have instability when that quantity goes to infinity. We can now calculate what makes this quantity go to infinity with simple algebra. And it's when the denominator goes to 0, which is when GC is equal to -1. Now, at this point at -1 is important, again, because it represents system instability when the transfer function goes to infinity. But what does any of this have to do with a neuron? This little block diagram, what does it have to do with the complex trees that we're trying to study? It turns out that we can take our complex system and approximate it with a similar block diagram. In our model overview with 4 or 5 components, we can approximate all of that as a linear model that behaves as a cascade of systems, the same systems that we recognized from earlier, cargo production, transport, and insertion, along with feedback, looping it all together. We can then compute the transfer functions, the output-input ratios of all of these, and plot them as a Nyquist plot. Now, a Nyquist plot is a graphical representation of the transfer function, the output-input ratio. Remember that point -1 that corresponds to instability? If a line on the Nyquist plot is close to -1, this red marker that we have here, then that system is less stable. We can see here that as we increase global gain, this kg, our system approaches -1. So each of these successive curves in these same colors are getting closer to that -1 point. They're becoming more unstable. So instability can be defined as a system's proximity to -1 on the Nyquist plot. There's a couple ways of measuring this. There's gain margin, which I've labeled as GM here, which is essentially the horizontal distance to -1. There's also phase margin, labeled as PM here, which is the angle of rotation to -1. So our next question is, what happens if we further increase kg? We're going to try that, and that's going to be this green curve here. In our simulations, we see that we've become fully unstable in C5, the green trace, and in the Nyquist plot, we've surpassed the -1 point, which also indicates instability. So using the Nyquist stability criterion, we can take a horribly complex model, reduce it, and quantify its stability. And this is how we do that in our theory. Next, we're going to take a look at accuracy. We're going to discuss both accuracy in synaptic potentiation and accuracy in synaptic scaling. Let's start with synaptic potentiation. To assess how well a neuron can potentiate its synapses, we're going to compare two equivalent neurites for a fair comparison between one neurite that's potentiated and one that's not. We're going to look at this over different internal pool sizes. Remember that this S minus I corresponds to the size of the internal pool. On the left, we have a small internal pool size, and the number of internal AMPA receptors dwindles with distance from the soma. On the right, we have a larger internal pool size, and there's sufficient AMPA receptors in both the proximal and the distal parts of the cell. Now, let's see how these examples fare with potentiation. On these bottom plots, we have percentage of full potentiation on the y-axis and distance from the soma on the x-axis. The shapes on these plots denote the degree to which the synapses were potentiated. The circles in the top lines correspond to doubling of synaptic strengths during LTP. The X's are 5-fold increases, and the triangles are 10-fold increases. From these plots, as we expect, we see that greater potentiation is more difficult for the neuron to sustain. And neurons with larger internal pools on the right here achieve a better percentage of full potentiation, better accuracy of synaptic potentiation. So given this result, this difference in accuracy, we ask ourselves, does the neuron have a small reserve of AMPA receptors like in B, or a larger reserve like in D? The answer turns out to be more like D. This is one of the earlier studies of AMPA receptor localization. The dendrites are all lit up, both in the spines and the dendrite shafts. And this is quite a bit of work for the neuron, producing and distributing all this material. But the upside is improved synaptic accuracy, as we've shown here. Next, let's talk about how we quantify accuracy of synaptic scaling. For this example, we'll start with a very simple toy neuron, a simple line of 4 compartments. Each of these compartments can be assigned some synaptic strength. So C1 is 1, C2 is 0.8, C4 is 1.2. C3 is going to start at 1 and then undergo LTP and be potentiated to 2. Here's what this looks like as a graph. We have time on the x-axis here, and halfway through our simulation, we're going to double the capacity of compartment 3, where the others will remain the same. If we run the simulation, we could see that the calcium reaches a steady state that is about halfway through our simulation, the calcium is going to experience a perturbation and then recover that same steady state. So homeostasis is maintained. Now for the actual synapses, that's this plot of GIs, all GIs reach a steady state, and then potentiation clearly makes C3 increase, which is the dashed line. All the other synapses experience a corresponding decrease after potentiation. And this is the essence of synaptic scaling. When synapses are potentiated, all other synapses are scaled such that the total synaptic activity, represented by CA, is scaled down. But the scaling does not occur equally. In red, I've plotted the mean values of unpotentiated synapses. In perfect scaling, all unpotentiated synapses would scale relative to this mean. And you can see that, for instance, the difference between these top two kind of goes away after scaling. This is imperfect scaling. So to calculate the quality of scaling, we can measure the percent error from this mean. For some synapses, there's no error, this one in the middle. For some, there's positive error, and for some, there's negative error. So to estimate the accuracy of synaptic scaling, it's sensible to average the absolute values of each of these, this bottom plot here. This is what long-term depression, or LTD, looks like. C3 is now decreasing to 0.5, and all other synapses are experiencing strengthening. And to ground us in reality, this is all pretty well established in experiments for cells undergoing LTP or LTD. So the point of these examples of LTP and LTD was to demonstrate that when a synapse is potentiated or depressed, all the other synapses in the neuron experience the opposite change. And in both cases, the scaling doesn't happen perfectly. We can compute a percent error from perfect scaling. And our measure of accuracy is the average absolute error of these. We're going to call that Q. Now let's see how Q, this mean absolute error, changes with morphology. So we're going to have a series of simulations in a simple cable model, and we have this measure of accuracy, mean absolute percent error, that I'm calling Q. We run the simulation for many potentiation ratios plotted on the y-axis and many internal pool sizes on the x-axis, and we're going to compute Q. So from this plot, we could see that error increases with stronger synaptic potentiation. That's expected. We also see that error decreases with larger internal pool size, which was one of the trade-offs. One thing we haven't mentioned is that all of this is highly dependent on morphology. In this case, we had a linear toy neuron. What if With a stellate architecture, like this one here, each compartment has much closer access to global control and the scaling signal. The result is much lighter blues, much lower errors for all of these equivalent conditions. And this is relevant when considering neurons of different shape. For example, a dentate granule cell might be more bushy and symmetric, like this on the left, which seems to have a pretty fairly compact morphology, whereas a pyramidal cell might be more linear and asymmetric, like on the right, which is comparatively more strung out. We're still working on the full morphology simulations of these two, but we anticipate the results when we control for neuron size might be pretty similar to these. So this is how synaptic scaling accuracy is affected by morphology. By now, we've talked about all these trade-offs of the triangle, and we now have a rigorous method of quantifying each of these three attributes. Let's try to put them together in a couple succinct figures. We're plotting two axes here, stability, which is our gain margin, as we saw earlier, and efficiency, which is the rate at which the system settles to steady state, like a time constant. This red line is drawn by repeated simulations with various kgs, or global gains, and this is the trade-off we expect. The low kgs have a high stability but low efficiency, and the high kgs are efficient but less stable. When we vary another property here, when we vary internal pool size, we can see that a small internal pool fills more efficiently, and this of course would come at the cost of synaptic accuracy, which is not depicted here. On the right, we're plotting on the same axes, stability and efficiency, and as we increase the amount of local control, as the line becomes more green, stability increases. And again, this would also come at the cost of synaptic accuracy, which is not shown here. So lastly, our model, simulations, and general framework allow us to make realistic predictions for the limitations of synaptic scaling. For instance, let's return to our full morphology example. We can now confidently say that for a human neocortical L3 pyramidal cell, that is receiving synaptic potentiation in the distal synapses for a total increase in synaptic strength of 14%, we can predict moderate accuracy, which we're saying is between 60 and 80% of full potentiation. We can predict stable scaling with minimal oscillations and a settling time of about 20 hours. It's worth noting that this is the best a neuron can do. If the neuron wants a faster settling time, something has to give. If the neuron wants better synaptic accuracy, it's going to be slower or less stable. So in our manuscript, which is coming out soon, you could look forward to everything I've talked about here, plus more simulations and predictions in real neuron morphologies. We have a pyramidal cell here, and we plan to do a similar analysis with a dentate granule cell, some Martinoli cells, and probably a few others. And we hope to make predictions regarding the stability, efficiency, and accuracy of scaling in those. In the end, going back to one of our questions from earlier in the talk, why is synaptic scaling so slow? Why does it take 1 to 2 days? Our framework shows that synaptic scaling is slow because it has to be, because neuron morphology constrains it. Neurons scale their synapses as fast as possible while maintaining accuracy and stability. As always, there's tons of people to thank and lots of organizations to be grateful for. My co-author and mentors, of course, the control group in the Department of Engineering at Cambridge for guidance and inspiration with the modeling and for presenting the stuff, and all the graduate student groups that I'm a part of. So thank you, and I'd be happy to take any questions.
Thank you so much, Adriano, for a great talk and perfect timing. We have 3 questions in the queue. I'll start by asking the first one, which is, how would these effects relate to structural plasticity?
Oh, that's a good question. We haven't done this in simulation, but well, first of all, structural plasticity, in my understanding, can mean a lot of things. When we change here, let me go back to a slide. When we change the capacity of a synapse, we're in a sense emulating structural plasticity at the local level. So this change in C in our model is taken to represent an increase in available slots in the actual synapses. So it's a change, it's an upregulation of many of the structural proteins in the synapse that can support AMPA receptors on the surface. So that's one kind of structural plasticity where we've changed the structure of some of the synapse to accommodate more AMPA receptors. So that's kind of taken into account here. Other kinds of structural plasticity where the neuron morphology changes and the actual branches and the trees change, we have not considered. We imagine that when that happens, this trade-off still exists and might be worsened, might be alleviated based on versus notice is that neurons that are more strung out, that require longer transport of the scaling factor, are going to be harder to scale. I hope that answers the question.
Yeah, you were cut off a little bit at the end, but I think we got the gist of it, so that's okay. The second question is, doesn't the ER catch most of the calcium flowing out to the spine? In such case, don't you think that the feedback signaling relies more on second messengers than calcium?
Yeah, that's a good question as well. So there's a lot of things that limit calcium transport. Like you said, the ER, also the spine neck is very restrictive. In our model, we aren't actually looking at calcium diffusion. So calcium can enter in the spines themselves. It can enter in the dendritic shafts themselves, and we're also saying that calcium can enter in the soma itself. So I think yes, that does happen. Calcium diffusion is limited by things like the ER, by the spine neck, and calcium concentration changes. We're also taking a calcium average over time as well, a spatial and temporal average. And our calcium dynamics are meant to represent the calcium and all the second messengers and the whole signaling cascade that changes with calcium concentration increase. That's a good question.
Yeah, there's a couple of comments on this question, so I'll read them to you. So one person says, yes, but the depolarization will cause calcium influx throughout the cell, including the soma. And the person that posed the original question says that's true, but in such case, can you call this process a calcium feedback? Because the amount of calcium that enters the soma is limited, and in this case, I understand that calcium levels may be higher than just regular somatic influx related to the depolarization. I don't know if that helps.
Sorry, I'm reading it now. Because the amount of calcium influx is limited. In this case, I understand calcium levels might be higher than regular somatic influx related to the depolarization.
I see that, yeah.
Yeah, this is true. The amount of calcium. So the amount of calcium influx into the soma also happens from inside the cell as well. There's internal calcium stores. And we expect calcium to fluctuate wildly because it is related in many second messengers for many other processes, not just synaptic scaling. Essentially, a lot of transcription factors, a lot of translation factors, and a lot of regulatory elements in the cell rely on calcium levels. So we do expect that to be a constant up and down. What we're assuming is that neuron excitability is related to that calcium concentration. And if we take a long time average and a pretty big space average in the soma for calcium, we expect that to correlate with neuron activity. I hope that clarifies that. But.
Yeah, one quick question. Have you looked at GABA A receptors?
We haven't looked specifically at GABA A receptors. We expect our model to generalize to many kinds of cargo that might be related, like NMDA receptors as well. But we haven't looked specifically at those. But these trade-offs we presume are fundamental, are for any ambiguous cargo that has to be regulated in this way over these types of morphologies.
Yeah, perfect. And then one last question. Does your model account for plasticity at one synapse, promoting plasticity at other nearby synapses?
In a sense, our model does, because our discretization isn't perfect. So the size of a synapse is quite small, and our discretization kind of groups some of these synapses together. So in a sense, when we're looking at potentiation, I'm going to go back to that first example here. When we look at potentiation of one of these synapses, it's really like a string of maybe like 50 microns. So in a sense, our model, when we're doing LTP in one synapse, it's really just like a row of 50 microns in the dendrite that's being potentiated. We haven't explored like heterosynaptic plasticity effects, where potentiation of one part of the dendritic tree might cause a decrease in the surrounding parts. So to answer that one, it's kind of a yes and no. It's kind of built into the model, but also kind of, there are other mechanisms that we aren't including here.
Excellent. That concludes the time for this talk. Thank you very much. Virtual applause, and we'll move to the next speaker. Okay.
Hello.
Hey, perfect. Adriano, we still see you. Okay, perfect. Okay, our next oral presentation is by Ada Johanne Ellingsrud. She's a PhD student at the Similar Research Laboratory, and her talk is titled Finite Element Simulation of Ionic Electrodiffusion in Cellular Geometries. Ada, take it away.
Thank you. Can you see my screen?
Yes.
Perfect. Thank you so much for the introduction, and thanks for having me here at the CNS 2020. Today, I will present a computational framework for finite element simulations of ionic electrodiffusion in cellular geometries. And I'll start out with some motivation for why we want to consider such models. Mathematical models for excitable tissue are commonly based on cable theory. And cable theory is based on the assumption that tissue can be represented in homogenized matter, which means that the extracellular space, the intercellular space, and the membrane lives in each point of the domain. And this is often a very good approximation, and cable theory has and will continue to provide a good insight into how excitable cells work. However, classical homogenized models are, for example, not able to capture how the morphology of the neuron or the extracellular space affect dynamics or represent local variations in ion channel distributions. And cable theory, at least in its standard form, also assume constant ion concentrations. And in many cases, this is again a very good assumption and a good approximation, as concentration changes are often limited by neuronal and glial uptake mechanisms that will strive toward maintaining concentrations close to basal levels. In the neural membrane, you have, for example, pumps and cotransporters, and also spatial potassium buffering by the glial cells. But again, there are several scenarios that involve dramatic changes in extracellular ion concentrations. On a large spatial scale, ion concentration changes are, for example, trademarks of several pathologies, such as multiple sclerosis, epilepsy, stroke, migraine, and cortical spreading depression. And the extracellular shifts will lead to changes in the neuron reversal potential and can thus affect the dynamical properties of the neurons. And as you can see here in the bottom right figure, that even slight alterations in extracellular potassium, calcium, and magnesium can affect the membrane mechanisms and firing of a model neuron. And also under non-pathological conditions, concentration-dependent electrodiffusive effects are hypothesized to be important in specific microdomains of the brain. And this is particular in regions where the extracellular space is small and confined. So today I will present a framework where we have removed both these simplifying assumptions. And this framework thus models tissue dynamics with both explicit modeling of the ion concentrations, or so-called electrodiffusion, and also in an explicit geometrical setting. And we do this by starting out with the previously presented EMI framework. And the EMI equation describes the dynamics of the extracellular, the membrane, and the intracellular electrical potentials with a geometrically explicit representation of the cellular domain. And that is why it's called the EMI model, because the extracellular, membrane, and intracellular dynamics. And in particular, we consider a domain with an extracellular region omega E and intracellular region omega I that are separated by an interface gamma that represents the cell membrane. And this is in contrast to homogenized model, where omega E, omega I, and gamma would live in each point of the domain, whereas here it's kind of separated out with this explicit representation. And in the IMA equations, we aim to find the electrical potential phi and the transmembrane potential phi m, such that 1 and 2 hold. And equation 1 here is the Poisson equation for the potential, where sigma is the electrical conductivity, whereas equation 2 state that the rate of change of the membrane potential is proportional to the current flowing across the membrane. And finally, we have interface conditions at the interface gamma, defining the membrane potential, and also state that we require continuity of the flux or the currents crossing the membrane. And this IMA framework only models potentials, as you can see. And what we have done is that we have extended this framework to also take into account varying ion concentrations. And we have done this by 2 main assumptions. The first one is that ions in the intra and extracellular domains are conserved and that they move due to either drifting electrical field or diffusion. And the second assumption is that the bulk is electroneutral. And this means that all net charge is associated as membrane charge and not resides spatially in the intra and extracellular regions. So the only non-zero charge density is that building up the membrane potential across the capacitive membrane or the cell membrane. And the rationale behind this assumption is that in neural tissue, the charge relaxation time, or the typical time scale that rho, which is the charge density, varies on, is in the order of nanoseconds. And that most of the local net charge density is resolved in nanometer thick layers surrounding the membranes. And by assuming electroneutrality, we don't really resolve these charge relaxation processes at the nanoscale, but rather assume that the charge density has reached a quasi-steady state where the charge density is derivative with respect to time equals 0. And this condition has been shown to be a good approximation on spatio-temporal scales larger than micrometers and microseconds. And the advantage is that this results in a more numerically stable framework for coarser time and space resolution. So for example, in comparison with PNP scheme, which actually resolves these charge relaxation processes, this is more stable for coarser time and space resolution, which allows for longer simulation on larger domains. And with these assumptions, we end up with the KNP IMA equations. And in this system, we aim to find the ion concentrations K, the electrical potentials phi, and the membrane potential phi m, such that the continuity equation hold, which is the first equation here, and the second equation stems from the electroneutrality assumption. And then again, the third equation state that the rate of change of the membrane potential is proportional to the current flowing across the membrane. And in these 3 equations, we have a flux density Jk for each ion species K. And this is modeled by a Nernst-Planck equation, where we assume that ions can move either due to diffusion or drift in the electrical field. And this framework holds for an arbitrary number of ion species K. And moreover, we have interface conditions on the interface gamma to couple the dynamics in the intra and extracellular domains. And these interface conditions define the membrane potential, continuity of total flux across the membrane, and how specific ion channels cross the membrane. And ion here is a functional describing the membrane mechanisms. And this is subject to modeling. So it could include, for example, ion channels, pumps, co-transporters, and so on. And ion typically depends on the membrane potential, some of the ion species, and the gating variable S, which again are governed by a system of ODEs subject to modeling. So the KNP IMA equations consists of a system of partial differential equations, potentially coupled with a system of ordinary differential equations describing the gating variables. And to solve this system numerically, we have applied a Mortar finite element method. And Mortar finite element methods allow for coupling of different types of variational problems posed over non-overlapping domains by weakly enforcing the interface conditions on common boundaries. So in our case, we couple the dynamics in the intracellular and extracellular regions weakly by IM, which is the variable living on the common boundaries, which is this interface mesh here, gamma. And I'm not going to go any further into the details in this numerical scheme, but I do want to mention some key advantages of a finite element formulation. First, independence of dimension. This scheme, the same scheme is applicable for 1, 2, or 3-dimensional domains, and also the handling of complicated interface geometries. One could easily swap this very simplified square 2D cell here with a more morphological, realistic cell in 3 dimensions. And to verify this numerical scheme, we have calculated the errors and the convergence rates during refinement in time and space. So what we have done is that we have defined an exact solution and then adjusted the boundary terms, source term, and initial conditions according to the solution so that we know what we expect the solver to yield when we run it. And in this way, we can calculate the error. And we have measured the error in the L2 norm and the H1 norm, and we observe rates of 2 and 1 respectively for the variables living in omega E and omega I, which is what we expected a priori. Finally, we have compared our KNP EMI framework to the original EMI framework. And we have chosen to do this by modeling a haptic coupling in unmyelated, tightly packed axon bundle. And such axon bundles can be found, for example, in the olfactory system. And it is hypothesized that these tightly packed axons can communicate through the very small extracellular space separating them via so-called haptic coupling. And as the KNP EMI framework includes varying ion concentrations, we wanted to test whether diffusive potential could strengthen the haptic coupling. And to do this, we have created a 3D mesh consisting of 9 axons separated by extracellular space. And we consider potassium, sodium, and chloride for the ions, and a standard Hodgkin-Huxley model is used for the membrane mechanisms. And here you can see the computational mesh, and here is a cross-section of the mesh with the 9 axons. And for the haptic coupling study, we consider 2 different scenarios. One where we only simulated axon A here in the middle to see how that affected the 8 axons surrounding it. And then one more extreme case where we stimulated all 8 axons here, B and C, to see how that would affect the 1 axon in the middle. And in figure A and B here, you can see the membrane potentials of passive axons with respectively 1 and 8 active neighbors. And on the y-axis, we have the membrane potential in millivolt, and on the x-axis, we have the time in milliseconds. And in figure A, you can see that with 1 active neighbor, we see a small haptic effect. The membrane potential increases slightly when a neighboring axon fires an action potential here at time 0, 25, and 50 milliseconds, which is this small bump in the membrane potential here. And with 8 active neighbors, depicted in figure B, the effect is more pronounced, but not strong enough to depolarize the membrane to the point where an action potential is fired. And you can also see the difference between the KNP EMI framework, which is in blue here, and the EMI framework in orange. And in the KNP EMI framework, we also see a persistent depolarization of the membrane that increases for each action potential fired. And this is due to the alterations in the Nernst potential. The Nernst potential will change for each action potential fired as potassium will be released into the extracellular space and sodium taken up by the cells. As the haptic coupling is inversely proportional to the extracellular conductivity, the conductivity is an interesting parameter here that we wanted to explore a bit further. In the KNP EMI framework, the conductivity is a function of the diffusion coefficients and the ion concentrations, whereas in the EMI framework, they are free parameter that you can set as you like. And by using physiological ion concentrations and diffusion coefficients, we get a KNP EMI conductivity of 2.10 Siemens-meter intracellularly and 1.31 extracellularly. And in the EMI framework, these are free parameters, so we can set them lower So that's what we did. We set the EMI conductivity to 1 in the intracellular region and 0.1 in the extracellular region. And then we repeated the same study with respectively 1 and 8 active neighbors. And we do see that in the case of 8 active neighbors, the ephaptic coupling is strong enough to induce action potentials in the neighbor, which you can see in figure F here. The KNP EMI framework I have presented here today is quite detailed, and consequently there are some challenges. There are more parameters that have to be set in the KNP EMI framework than in simpler models, which will cause more uncertainty. And moreover, running simulations using the framework is fairly computationally heavy. So homogenized models and models with constant ion concentrations are still very much relevant. But for certain phenomena, in particular related to how ions affect dynamics at a single cell level or for a few cells, for example, how the morphology of the cell or the distribution of ion channels locally affect dynamics, this level of complexity is needed in order to capture the right dynamics. The work I presented here today is done in collaboration with Andreas Solbro, Gaute Einevold, Geir Halnes, and Marie Rognes. And it is also summarized in this paper published in Frontiers in Neuroinformatics. And moreover, the source code is free and openly available and published online. And you can also find a link to the code in this paper if you would like to check it out. Thank you so much for your attention.
Thank you so much, Ada. That's a great talk, and thanks for sharing the resources. We have 2 questions in line. The first is from Alexandra Kerry Chelsea Calimbio. I hope I didn't botch it too badly. The question is it accurate to assume that this new framework corrects mainly for ion diffusion and pumps as opposed to synaptic transmission compared to Ralph's equation?
Yes, I would say that this framework is mainly for considering the effects of ion diffusion. So it's kind of at this mesoscale. So you do have even more detailed frameworks to consider if you want to look at kind of a patch of membrane or kind of very close at specific channels. Whereas this framework is good for capturing dynamics due to diffusion on kind of a single cell level, or if you have kind of a few cells.
Excellent. We have another question from Jonas Verhellen. Have you considered finite volume schemes?
No, we have not. We have only considered a finite element scheme. That is partially because the lab I'm working at has developed software for finite element simulations. So that's probably a bias there. So that's the only thing we have considered so far.
Okay, and probably there will be more questions coming in Neurostars, so definitely check there. You have a lot of appreciation for seeing the code shared and a lot of applauses, so I'll join them. Thank you very much.
Thank you.
Thank you. We'll wait a second, and in the meantime, we'll ask Heidi to come to the stage. So, great.
Hello.
Yes, all good. You're okay with sharing your screens?
Yes, I will do it now.
Okay.
Select window. Can you see my slides?
Yes, perfect. So I'll finish your introduction. So Heidi Teppola is a doctoral student and Faculty of Medicine and Health Technology at Tampere University. And her oral presentation is going to be titled Discovering Synaptic Mechanisms Underlying the Propagation of Cortical Activity a model-driven experimental and data analysis approach. Take it away.
Hello, everyone, and thank you for having me here in CNS 2020. My name is Heidi Teppola. I'm a biochemist and neuroscientist in computational neuroscience lab at Tampere University in Finland. And I'm going to talk to you about our novel results, how synaptic mechanisms contribute to spontaneously occurring synchronous cortical network activity in vitro, and how the experimental results can support the development and fitting of spiking network models. Spontaneous synchronous activity is described as a hallmark of developing neocortical networks and functional maturation of sensory cortices during the late embryonic and early postnatal stages. This activity is thought to prime the network for incoming sensory information, and it is known that the network activity forms in parallel with structural and synaptic connectivity, and that abnormalities in the process lead to neurodevelopmental disorders. Here you can see the timeline of developing rodent cortex from late embryonic until early postnatal states, with two examples showing the spontaneous activity patterns recorded in the form of network oscillations from postnatal cortical slice in vitro and spindle and gamma burst in somatosensory cortex in vivo. In mature dissociated cortical networks, the landmark of spontaneous activity is the repetitive emergence of short period of intense spiking, separated by longer silent steady interburst intervals. Each propagating burst has a unique spatiotemporal pattern in which neurons at different locations fire spike trains at different delays relative to each other. Bursts are shown to be initiated from localized areas in these networks. Cortical network bursts are known to be driven by the excitatory synaptic transmission, which is primarily mediated by the action of glutamate on two types of glutamatergic AMPA and NMDA receptors. Inhibitory synaptic transmission is mediated by the action of gamma-aminobutyric on GABAergic receptors. GABAergic signaling and interneurons are crucial in developing neocortex. Both AMPA and GABA receptors express fast and NMDA slow ion channel kinetics. Excitatory and inhibitory synapses are shown to be evenly distributed along the dendrites in networks at 3 weeks in vitro. Although cortical activity is a well-known phenomenon in developing networks, the contribution of synaptic receptors to the network activity is not well understood. Because the mechanisms underlying early network activity is not well understood, it's necessary to construct realistic in silico models fed with experimental in vitro data. These data-driven models help us to understand how synaptic and cellular properties affect the network burst activity. By constructing models and modifying their parameters, we can explore which underlying mechanisms are necessary to reproduce the different type of activity regimes observed in vitro recordings. And previously, the spiking network models have been developed to study the burst dynamics, burst initiation mechanisms, and connectivity on network activity. In our study, we present the first data-driven network modeling approach that uses microelectroarray data as a training and test data to study the impacts of synaptic and cellular mechanisms on network activity in rodent networks in vitro. The aims of this study are to examine the contribution of AMPA, NMDA, and GABA receptors to initiate, propagate, and terminate the cortical bursts with microelectroarray recording technique combined with pharmacology in rodent postnatal networks in vitro, and to develop this model-driven data analysis workflow to support the development of data-driven spiking network models in silico. I started this study by extracting cortical neurons and astrocytes from the forebrain of postnatal rats. I cultured and maintained the cells on microelectroarray plates until 3 weeks in vitro and recorded the extracellular network activity using 60 electrodes simultaneously over many hours. For more details, you can find from our recently published paper, Teppola and others, from Frontiers Cellular Neuroscience. I recorded the network-wide activity under several pharmacological conditions of receptor antagonists, I performed two types of experiments: acute, complete blockage of each receptors, and gradual suppression of receptors. In acute application, first one of the excitatory pathway is blocked, followed by blocking of inhibitory pathway. In the gradual application shown here, I blocked either AMPA or NMDA receptors. Data analysis of microelectroarray data was conducted in a way that it supports both the neurobiological questions and the fitting and validation of computational models to accurately produce the experimental results. Here you can see 60 electrodes in a grid format that were used to record extracellularly the action potential waveforms from each electrode. Once we have recorded the network activity, we detected the network bursts. Then we computed burst measures from burst profiles, such as maximum firing rate, burst length, rising and falling phases, and burst sizes. For model fitting purpose, we used two measures the distribution of burst size and length, meaning in each pharmacological condition. And for characterizing the similarity between spatiotemporal propagation patterns, we computed the first spike time relative to the first onset for each electrode, and the similarities as the correlation coefficient between pairwise spike time different matrices in each of the patterns. We also developed spiking network models that were fit to experimental data I have recorded. We constructed a network model by randomly connecting spiking point neurons with synaptic models. The synaptic models contain the short-term presynaptic dynamics and glutamatergic and GABAergic synaptic receptor conductances on the postsynaptic side, as considered in the experimental study. My colleague, Jugoslava Acimovic, built a new procedure where this model was automatically fitted to the experimental training data, using a computational intensive genetic algorithm to ensure that the models exhibit same activity as seen in vitro. We fitted the model cell parameters, synaptic receptor conductances, and the parameters for released synaptic resources. In each of the iterations, the fitting algorithm, a number of copies of the model were generated with different model parameters. Models were simulated and the network burst detected, and the quantitative measures of burst profiles, such as distribution of burst length and sizes, were computed. Comparison between in silico and in vitro test data allowed selection of best models. This protocol allowed to examine the effect of cellular and synaptic mechanisms on cortical network activity in silico. For more details, you can see our paper from Acimovic and others under submission. Here you can see our novel experimental results showing that the network robustly emerged in the presence of either AMPA or NMDA receptor antagonists. Both blockages significantly decrease the total firing rate of the network activity. However, the total firing rate was decreased more by NMDA than by AMPA receptor blockage, which is an indication that NMDA receptors have also an essential role in this cortical burst activity. Next, we computed the burst frequency that significantly decreased in the AMPA receptor, but not in the NMDA receptor blocked networks. These results indicate that AMPA receptors play a critical role in the initiation of network bursts. Then we characterized the burst measures from burst profiles, part of which were used to fit and validate the computational models. Interestingly, burst length and size increased after the AMPA receptor blockage, which shows the important role of NMDA receptors in maintaining bursts. Vice versa, the blockage of NMDA receptor significantly decreased the burst length and burst size, indicating that AMPA contributes to the early and NMDA receptor to the late phase of the bursts. We further studied the role of inhibition in network activity. Results show that when blocking the fast inhibition, total firing rate significantly increased in AMPA receptor-mediated networks, but not in the NMDA receptor-mediated activity when disinhibited. This result suggests that AMPA and GABA receptor with fast channel kinetics have a strong interaction in the network activity. Sorry, we also saw that the interspike intervals became much shorter in AMPA receptor-mediated activity than in the NMDA receptors, which is another indication. Here I'm going to show you the obtained result from in silico study. So we constructed the separate neuronal network model to each of the 5 experimental conditions: control, AMPA blocked, AMPA and kappa blocked, NMDA blocked, NMDA and kappa blocked. So each of the column is assigned to different pharmacological condition, showing model fitting precision for the selected burst measures, which are distribution of burst length and size. The blue line is the distribution computed from the experimental data, and the red line represents the pooled in silico data obtained from repetition of the model fitting. And the value in each plot shows the goodness of fitting, and if the value is less than 0.2, it indicates that model fitting succeeded. So fitting succeeded for almost all of the condition, although NMDA and kappa blocked condition, it was more challenging to achieve good results. Please see more detailed description of results in our paper, Acimovic and others, that is under submission. Finally, we studied how fast the network is recruited when the spatiotemporal patterns propagate in these cortical networks. And we found that NMDA receptors slow down the recruitment of neurons, while AMPA receptors rapidly recruit neurons to the network. We also analyzed how similar the spatiotemporal patterns are. And interestingly, we found out that when gradually suppressing AMPA receptors, similarity between the network burst patterns significantly increase. While gradual NMDA receptor blockage did not have an influence on the similarity of spatiotemporal patterns, indicating that AMPA receptor increased the diversity and richness to spatiotemporal patterns. Finally, I would like to conclude our 2 papers presented here. So first of all, it's very important to study and model the contribution of synaptic receptors to cortical network activity, since we know their important role in learning and memory formation. So in the experimental study, we found out that AMPA receptors have dominant role in the initiation of burst by rapidly recruiting neurons to networks. NMDA receptors maintain the elevated network burst, and GABA receptors strongly inhibit the AMPA receptor-mediated spiking. Furthermore, a profound interplay is found between the fast excitation and inhibition, with richer and more diverse propagation pattern than its absence. Additionally, we have constructed new data-driven network modeling approach that uses microelectro data as a training and test set to study the impacts of synaptic and cellular mechanisms on cortical network activity. And the optimal model reproduces with remarkable accuracy the dynamics of network burst in most of the pharmacological conditions. I would like to acknowledge the people who have made this work and collaboration possible, including Professor Marja-Leena Linne and my colleagues, Dr. Jugoslava Acimovic and Dr. Tuomo Mäki-Marttunen. Special thanks to all members of the Computational Neuroscience Lab, and thank you very much for your attention.
Thank you so much, Heidi. We have time for a few questions. One of them says, did you connect the spiking network model with the random uniform distribution?
Yes, we had a random connectivity in this study. Exactly. So we have another paper from Tuomo Mäki-Marttunen and others, which is studying the connectivity rules in this network burst activity. But in this study, we use random connectivity.
Wonderful. And then one other question. Have you looked at the role of an NMDA receptors with different subunit composition? For example, an R2A versus an R2B containing NMDA receptors.
Yes, we have not looked at that data in these works, but it's very interesting question, and I think it's a very good idea to experiment that. Since it's
Then one other question, you mentioned astrocytes. Have you looked at their role in your model?
in this model which is coming out now, we don't look at the astrocyte, but we have another article in preparation where we also consider the role of astrocytes in the network activity. And from yes.
Wonderful. So one question: did the models show the same effect that you saw in the experiments, or have I missed it?
Yes, so most of the experimental conditions or pharmacological conditions showed, and so I can go back to this slide. So with NMD and kappa blocked condition, we had some challenges, and this is most probably due to the lack of NMD and kappa when there is only AMPA conductance, and that makes it difficult to reproduce the results for burst size and burst length. So there are either very high spiking here, and then the length is fine. But in case you want to have the burst size accurate, then the burst length disturbs from that. So I think this is due to the AMPA, short AMPA conductance, time constant for the AMPA conductance. Is there still some questions?
No, there's actually, we have answered everything. You have many rounds of applause. I'll join them. And thank you so much for sharing your beautiful work.
Thank you so much.
There's one last question. We just arrived.
Ready for it.
Since we have a couple of minutes to spare, how do you know whether it's GABA or AMPA? Did you do any staining?
Yeah, that's a good question. So I haven't done. Do you mean like if the GABA is depolarized in the networks, or?
It seems. I mean, if I'm interpreting the question correctly, it's just a staining question. How do you know what receptors you have and where they are?
I have not stained them, so I was showing here the. Yeah, that should be done as well. That's a good point. So, but I know that the GABA receptors in these networks, 3 weeks in vitro, they are already hyperpolarizing, so they are not depolarizing. As known that there is this GABA shift in the early network activity, but that has happened already in these networks. So.
Okay. Yeah. Perfect.
Yeah, thank you.
I think that concludes the questions we had.
Yes.
Thank you very much.
Yeah, we can continue discussion in Neurostar. Thank you.
Definitely we'll find the questions there. Great work.
Thank you very much.
Thank you. Bye-bye.
Bye-bye.
Okay.
Hello.
Okay, perfect. We are 1 minute ahead of time. So if you want to check your slides in case, we can set them up.
I will look at them. My connection was lagging a little bit, so I hope that's fixed now.
Wonderful.
Sorry,
That's okay. Yeah, we have 40 seconds to spare.
somehow it's not getting my... I'm going to try again. I'm sorry, it's not letting me share my screen at the moment.
Okay. If worse comes to worse, you can.
I can exit and come back.
You can do that. If it gets really bad, you can also email me your presentation and I'll share my screen.
No, I will try to reload. Yeah, I will try to reload and see what happens.
Of course. Yeah, we'll wait for you.
Yeah. Sorry about that. I don't know what happened.
That's okay. Okay, our next speaker is connecting. Just a couple of seconds. Okay, she's back.
Here I am again. Okay. Let's try to do it. Okay. Are you seeing my slides now?
We don't. Okay, yes. Perfect. Yes. Success.
Finally.
Okay, so we can get started. The next speaker for this session is Paula Sanz-Leon. She's a senior research officer at QIMR Berghofer, and her oral presentation is titled Neural Flows, Wave Velocities and Singularities in 3D plus T brain data. Take it away.
Yes. Thank you very much. Well, thank you to the organizers for selecting my work for this oral presentation. To the web team, you are on fire behind the scenes, and it's been a pleasure working with you so far. And also, thank you to every one of you on the other side of the screen for listening to this presentation. So I'm going to walk you through a general overview of a toolbox I've been developing for the last 18 months, perhaps. And this toolbox we developed to estimate wave velocities and singularities in three-dimensional space that evolves in time, in particular focusing on neuroimaging data like functional MRI and MEG. So this talk is going to be very simple. It's split in two sections, so background and then an overview of the toolbox and some of the applications and preliminary results that I have. So background, waves in the brain. We know, and now there is a lot of evidence that neural activity exhibits constantly evolving, very complex spatial temporal patterns. And the switching between these patterns that sometimes we call brain states, can explain large-scale functional reorganization. And what do we mean by this large-scale functional reorganization? Well, actually, when your brain activity actually reconfigures because you're doing different tasks, for instance, like speaking now, or maybe looking at some window, or answering some SOS message from some speaker that cannot put her slides up. So why is it so important that we understand these patterns in the first place? Well, we think that if we improve our understanding of the dynamic interaction between all pairs of brain regions, for instance, we will better understand complex cognitive functions. Yeah. And at the level of the whole brain, these brainwaves have been observed in 2016 by Mueller and colleagues. They saw these rotating patterns in the brain during sleep. And last year, James Roberts and colleagues predicted these metastable brainwaves. So this highly structured activity that is switching over time. So how do we characterize these complex spatio-temporal patterns at the level of the whole brain? Well, we have adopted in particular some tools from fluid dynamics. We are going to make two assumptions. The first one is that neuroactivity is an active fluid, and by active we mean that there is some energy, free energy, that is transformed into work, and this process is actually powered by metabolism in the brain. And there are also some activity particles that can be tracked over space and over time. And this is what we call brain dynamics analyzed as neural flows. So let's have a look at what this neural flow representation actually is. We have some measurement points here. I hope you can see my, can you see my pointer? No.
Not at the moment.
No.
Oh, now yes. Now yes.
The big arrow. Okay. Yeah. I just wanted to make sure you can actually see it. So we will have some measurement points. These are fixed locations, and this could be sensors, for instance, of EEG, or it could be parcels in parcellated schemes for functional MRI. And if we have 2 time points of this data, we can calculate some sort of vectors. And these vectors will indicate how fast and in which direction activity is moving. Yeah, so each one of these arrows is a flow or velocity vector. And in this talk, I will speak about flow and velocity like interchangeably. So it's in units of meters per second. The second representation is in terms of activity particles, and you can see that we can track them over time, and we can track this trajectory that they will make paths or functional paths in the brain. And I want to acknowledge that there are some other tools as well that use the same fluid dynamics framework, but they operate in 2 dimensions, or in images that are 2-dimensional and that evolve over time. And so here are some of the important references: NeuroPath in 2018, O10, it's MATLAB and C++ in 2017, and WAVE, which is a collection of functions that Lyle Mueller published with the eLife paper of the rotating waves. And we can see that we have some activity that then is transformed into these vector fields, and all of them, they share this approach. And in particular, we are looking at these nice points here that are called critical points or singularities. And these are important because they are a sparse representation, actually, of a very complex spatio-temporal pattern. So what aims do we have here? We want to provide a representation of brain function that accounts for the spatial embedding of the brain, but also that we can track information flow encompassing geometrical and temporal aspects, because so far many analysis methods either focus on temporal aspects, maybe spatial, but never at the same time. And we want to produce a sparse representation of complex spatio-temporal dynamics. So what do I have to do? Well, estimate wave propagation velocities, detect some spatio-temporal critical points. Surely this should be easy, I told myself 18 months ago. but it's in 3 dimensions on time. So if I add one extra dimension, how that can be? Well, here I am. This is a paper in preparation, and hopefully will be submitted and published soon as well, with the description of the toolbox. And I owe you the user manual, but it will also come soon. So let's get to it, to the toolbox. What can you do with this toolbox? Well, a typical workflow, it's like this. We have some data. In general, this data comes it comes from some scattered measurement points. As I was saying, source reconstructed MEG or EEG or parcellated fMRI. So we need to interpolate. Then the toolbox will estimate your flows. We can use this using the amplitude or the instantaneous phase as well. We provide different methods to do this. We'll get this representation in terms of flow fields. We will detect 8 types of 3-dimensional singularities, from foci or stable fixed points, spiral fixed points, and saddle points. We also have a representation of streamlines, and we trace these streamlines over space and time. And these streamlines are giving us an idea of these functional pathways that form and dissolve in the brain. And last but not least, we provide analysis methods for flow decomposition to actually make sense of this high-dimensional information. And we track these singularities, as you can see over here, over space and time. Here is the x-axis, y-axis, and z-axis. We see a cloud of points here that is indicating a very complex spatio-temporal pattern. Then all singularities disappear, and this is actually a traveling wave at the level of the whole brain. Then it gets like an anchor point, and it transforms into a rotating wave and some other complex patterns. And I can tell you this because I've been looking at these plots for a long time now. So this typical workflow led to a modular and simple architecture of the toolbox. It has some input-output operations for partial reading and writing, because we are operating on really heavy data sets and producing really heavy data sets. So this toolbox is tailored for high-performing computing clusters, but also you can run it on your laptop because it doesn't use a lot of memory. It's simply that it abuses a little bit of reading and writing operations. We have the core module that actually maps to this workflow, process data, estimate flows, identify singularities, streamlines. We do some analysis, and we also provide visualization. So the input datasets that you can use, it can be this unstructured, as we call it, so these scattered points in 3-dimensional space, or it could be structured, actually, so voxel data as well. The configuration file to orchestrate absolutely everything that you can do with this toolbox and say to the toolbox, well, do this, but not this other calculation, is a configuration file in JSON format. And the reason to select JSON format is because it aligns a little bit with some efforts regarding data processing pipelines like fMRI prep. And we hope at some point that we can include our toolbox as part of some analysis pipelines, more complex analysis pipelines. So it's starting point here. So flow estimation, I was going to give you some really big equations here, but essentially we use a very well-known method called Horn and Shank from computer vision, an optical flow method, and a definition of phase gradient. So activity now is represented as a flow field. And we can see here, this is a rotating pattern, but the flows are changing. So there is some acceleration too. And we can understand these velocities and acceleration in terms of energy. If this gets a bit too complicated, we are looking at some slices here of these flows, and we're tracing these streamlines or functional trajectories. And we can see that these flows have also some complex patterns, but some structure and some simplified structure emerges from this. In particular, you can see here some saddle regions, meaning that any activity particle will come here and then will be diverted, right? This is just 2-dimensional slice for the sake of presentation, but this is actually, we have this in 3 dimensions. The color here represents like activity of high speed, so things are moving really fast, while the dark blue is activity is moving very slow. Because this is very complex, what we want to understand, for instance, is some of the most common flow patterns of activity that we have. And so now we use something like singular value decomposition. Singular value decomposition. Sorry, I was just having a look at the chat to see if there was anything going on there. So flow decomposition, we use singular value decomposition again, and we find that with 4 flow modes, we can capture all the complexity of the flow fields. What this means is actually, with respect to time, one of these patterns will actually contribute the most to the flows and therefore to the complex spatio-temporal pattern that we observe. And as a side effect, we can also estimate conduction speeds. And you have here a distribution of speeds in meters per second. So I mentioned about some singularities, and now you can have a closer look at them. So we are tracking them over time and space. And these are the type of singularities that we have. I'm showing 4, but I mentioned there are 8. And here we have some, what the toolbox produces is a summary of singularity statistics, and this comes to the sparse representation that we wanted to obtain, and I mentioned at the beginning. So we have nodes or number of regions of interest, and we have time. So each of these color points, I will show you a Zoom version soon. It's tracking what type of singularity is going on in that part of the brain. And we produce some other statistics here, like occupancy, so how many frames each singularity persists, and for each time point as well, how many of these singularities appear and disappear. And again, for each node here, we are estimating how much time this node spent being, for instance, a spiral saddle or spiral sink or spiral source. Here is a Zoom version. You can see it, I hope, a little bit more clear. So singularities are changing, and so this indicates changes in stability of this dynamic. So does it work with real data? Are there really brainwaves and singularities lurking in neuroimaging datasets? Well, spoiler alert, there are actually, because those summary statistics I showed you earlier are from actually an fMRI dataset, and I will show you now. Hopefully it's not too jumpy. So these are brainwaves in functional MRI data, and these hairy-looking movies are actually the functional pathways that form and dissolve over time. And you will see at some point some of these pathways going backwards when you have something going into the visual cortex. And it actually makes sense and looks very complex because this data set was acquired during movie watching, actually, not during resting state or a particular task. So we can trace where activity goes with these streamlines, and we can see that they flow from a particular part of the brain into another, or they reverberate, which are these rotating patterns that sometimes you see here. Below, I showed this flow mode decomposition into 10. As I say, I need more modes here to capture the complexity of fMRI activity, because, again, it was during naturalistic movie watching, so lots of sensory modalities are engaged. And I present some of the singularity of this sparse representation. Maybe for you it doesn't make a lot of sense, but we are going to use these into classifiers to classify these large-scale patterns in a more compact way. So if you can't get enough of this, and I think I'm running out of time, we have a poster. There is also a YouTube video in case that I don't make it to 3 a.m. here in Australia to be there, but I will do my best. And also poster 172 on brainwaves in resting state in MEG data. And I also invite you to a workshop if you like spatio-temporal dynamics, both modeling and analysis. I invite you to our workshop on Tuesday, actually. With that, I will thank my collaborators, James Roberts, Leonardo L Gollo, and Stuart Nock, both for guidance, discussions, and also for getting a lot of bugs, annoying bugs in the code. And if you have any questions, well, I will answer now. You can contact me if you have bug reports, feature requests, help, or whatever, you can see a pre-release of the code at this address, and opening new issues are always welcome. So thank you for listening.
Thank you so much, Paula. That was a beautiful talk. We have 5 questions for you. The first one is, how do you deal with the folding of the cortical surface? Could folding induce artificial singularities?
Okay, the simple answer to that is that I don't deal with the folding of the cortical surface. Actually, we already lost that information the moment the input data set already lost that information once it's heavily discretized, like parcellated functional MRI or source reconstructed MEG. I do have a working implementation that operates on the cortical surface. The methods are a bit more tricky there. And could the folding induce artificial singularities? Yes, it could be, and that's why these methods that I'm mentioning, that are not the primary focus of the toolbox at the moment, require a high-resolution surface to avoid this. Just a brief mention, a surface is a 2-dimensional object, and so it requires to re-implement a bunch of methods that I have implemented for 2 dimensions. But yeah, so thank you for that question.
Perfect. Next one, thank you for this nice work. I'm wondering how you quantitatively test or validate the estimation of the flows. Also, the waves can be positive and negative amplitudes. So shall we separately do statistics on them?
Could you repeat that question? I got
sure.
a little bit. Yep.
Yeah. I'm wondering how to test quantitatively or validate the estimation of the flows. And a second part of the question is, can the waves be, since the waves are both in positive and negative amplitudes or directions, can you do separate statistics on them?
Okay, I will try to answer that in 2 parts. The first is about validation. Yes, so the toolbox has validation, and these results will be shown in the paper. We actually created 3-dimensional waves with closed forms, like from a function of a wave, and spiral waves and traveling waves and so on. So we know actually the propagation velocity, and we estimate this, and yes, the results are there and they're accurate. Of course, there is some error when we calculate interpolation, or the moment we start having a really large spatial sampling, then yeah, the error will grow. But we have validated that the velocities we estimate are correct. In that sense, the same with singularities. We generate singularities and we pass them to the classification method, and yeah, it correctly classifies this. As for the second part of the question, sorry.
it's very late. Yeah, sure.
Separate statistics. For separate statistics for positive and negative values. I guess you could do that, but like a wave, the amplitude, these flows actually look at spatiotemporal derivatives. So I'm not sure why you would do like separation between positive and negative values, but sure, you can do that. The toolbox doesn't really know what the quantity that you are giving us amplitude is. So I guess it depends what you want to get out, and based on that, the interpretation maybe of these speeds or velocities might change a little bit, but you can.
Okay, very quickly, how well does this method work with highly noisy data? And what's the difference between Horn-Schunck method and other optical flows algorithm like FarmBack?
Okay, excellent question. How well this method works? So it's a numerical method. So it will work insofar that you put some numbers in the computer and it will give you some other numbers. right? But yes, you're right. With very highly noisy data, the convergence of the optical flow method might break down a little bit, and yeah, you will introduce noise in these flow fields. So because the Horn and Shank method relies on at least twice differentiable data, both in space and time. So you do need smoothness. And the difference between the Horn and Shank and other optical flow methods, well, Horn Shank is called global method. It means that there are some smoothness properties or parameters that remain the same for all areas of the space that you are treating or all areas of the image. But there are other ones that are called local, so you can change these smooth constraints over space. That's one of the main difference, like with the algorithm like Farnibach. But one of the reasons we picked Horn and Shank is because it's been thoroughly validated and used in the context of neuroscience data or neural data. It's been mathematically proved that it converges for n dimensions, and it's easy to implement and easy to understand. Sometimes I pick something that's easy to implement and understand for others over some obscure but super fast algorithm. So.
Wonderful. We have 2 more questions, but I would say probably a very quick answer to one, and then we can move them to Neurostars.
Yep.
So what are the requirements? What requirements do you have for the data to use in these analyses?
Requirements, I require the locations of measurement points or locations of brain regions and the data. The input is a MATLAB format, and I don't require anything else because I didn't want to overhead of having to handle different formats. So that comes from some other toolbox, let's say, and you put it into a toolbox. It has to be smooth too. So.
You did such a wonderful job that I don't want to miss the last one now. Are the commonly observed flow types optimal in terms of metabolic energy usage?
Ooh, tough question.
The last one is always
the answer is I don't know because I haven't looked into that, but that's a really nice question, and I would like to find out, or maybe you can use the toolbox and find that out. I would be happy to see that.
Okay, the conversation can move on to Neurostars. Thanks everyone for a great session, and stay tuned for the remainder of the meeting. Thank you, everyone.
Thank you. Bye.
Bye-bye.